So we're going to be continuing from where we left from. Remember, we had talked about the types of glycolysis, and we're just going to discuss now what are the uses of NAD+. But before we talk about that, let's look at a few exceptions in this process of glycolysis. So exceptions, we have some organs that always follow anaerobic glycolysis. For example, the brain, retina, and erythrocytes. So these ones always follow anaerobic glycolysis, even in presence of oxygen. So why do they follow anaerobic glycolysis even when there is oxygen? This is because ATP production is 100 times faster in anaerobic glycolysis. So now let's go back to our question here. What's the use of NAD+? Which takes us to the next page. So uses of NAD+. NAD+ is used in the continuation of glycolysis, without which glycolysis is going to cease, it's going to stop. So NAD+ is used in the continuation of glycolysis, without which glycolysis ceases. So for example, this NAD+ is going to be used in this reaction in glycolysis, where we're going to convert glutaraldehyde 3-phosphate to 1,3-bisphosphoglycerate using the enzyme glutaraldehyde 3-phosphate dehydrogenase. And you see here we're using the NAD+, and which is going to be reduced to NADH. So our next question will be phases of glycolysis. So we have two phases in glycolysis. We have the priming phase and we have the energy yielding phase. So the priming phase is where there is energy expenditure. So -2 ATP. And the reactions involved in this priming phase are reaction 1 of glycolysis, which involves glucose being converted to glucose 6-phosphate using the hexokinase enzymes in an irreversible reaction. And here we can see that we are using ATP. ATP is converted to ADP. Yes, and then the next reaction in the priming phase will be reaction 3, where fructose 6-phosphate is converted to fructose 1,6-bisphosphate, also in an irreversible reaction, and then ATP is being converted to ATP, meaning we are using ATP. So how did we get fructose 6-phosphate? After we've converted glucose to glucose 6-phosphate, we are going to convert glucose 6-phosphate to fructose 6-phosphate using the enzyme hexokinase isomerase. So the next phase after the priming phase will be the energy yielding phase of glycolysis, where we're going to form 4 ATPs plus 4 ATP. So this is the energy generation phase. Also, in this energy generation phase, we're going to produce two NADH molecules. So the reactions for the energy yielding phase would be reaction 6 of glycolysis, where glutaraldehyde 3-phosphate is converted to 1,3-bisphosphoglycerate catalyzed by the enzyme glutaraldehyde 3-phosphate dehydrogenase. And here we're using NAD+. You can remember our reaction above here. Yeah. And here we're using NAD+. And this 1,3-bisphosphoglycerate is a high energy compound, which we're coming to just right now. The other reaction involved in the energy yielding phase would be reaction 7, and this is where we're going to talk about this part, high energy compound. So this reaction 7 is a substrate level phosphorylation, where we're going to convert 1,3-bisphosphoglycerate, which is a high energy compound, to 3-phosphoglycerate catalyzed by the enzyme phosphoglycerate kinase. And here ADP is going to be given an inorganic phosphate, and we're going to form ATP. And here we're forming 2 ATP molecules. This reaction is ×2 because you remember in glycolysis, after fructose 1,6 bisphosphate, it's going to be converted to two molecules catalyzed by aldolase. We're going to form glutaraldehyde 3-phosphate, and we're also going to form dihydroxyacetone phosphate. But glutaraldehyde 3-phosphate is the one that is favored. So dihydroxyacetone phosphate is usually converted to glutaraldehyde 3-phosphate using the enzyme thiose isomerase, and so we're going to be proceeding with glutaraldehyde 3-phosphate till the end, but we remember to multiply by 2. So let me show you what I'm trying to say just a bit. Oops. Here we go. This is what I am trying to say. Fructose 1,6-bisphosphate is usually converted to glutaraldehyde 3-phosphate and dihydroxyacetone phosphate, catalyzed by the enzyme aldolase. However, glutaraldehyde 3-phosphate is usually the favored one. It's the loved child. So dihydroxyacetone phosphate is usually converted to glutaraldehyde 3-phosphate using the enzyme thiose phosphate isomerase. And now from then on, we're going to be proceeding with glutaraldehyde 3-phosphate till the end. We're going to be forming pyruvate, but every time the products, we're going to be multiplying them by 2. So I hope that is now clear. Yeah, so we can continue with our next question. Yeah, so other reactions were in the energy yielding phase. We've talked about the reaction 6. We've said that in this energy yielding phase we're forming 4 ATP and also NADH molecules, right? 2 NADH molecules. So for the NADH molecule, we've already formed it here. These are 2. Remember this, this is times 2. So we've already dealt with this one. What about the 4 ATPs? That's what we're going to now. So reaction 7, which 1,3-bisphosphoglycerate, remember it's a high energy compound. It's going to directly donate one of its phosphates to ADP, and we're going to form ATP. And you remember the story of multiplying by 2? So we have our first 2 ATPs. The next reaction is reaction 10, which is also a substrate level phosphorylation, where phosphoenol pyruvate is converted to pyruvate, catalyzed by the enzyme pyruvate kinase. And then we form, and then phosphoenol pyruvate donates its phosphate to ADP, and we form ATP. We're multiplying by 2. So this is our next 2 ATPs, and 2 plus 2 will give us 4. So the total ATP is 4 ATP molecules. Our next question will now be, what is substrate level phosphorylation? And we're going to say that substrate level phosphorylation, we're going to say that substrate level phosphorylation is the formation of ATP from substrates. These substrates are high energy compounds, and phosphate group is directly transferred from the substrate to ADP to form ATP. Yeah. So for example, reaction 7 and reaction 10 of glycolysis, which we've just seen above here. Yeah, so that's going to serve as our second video of questions, and we're going to have another one to just continue and finish up.